Hello there. Welcome back to Midnight Ocean. I'm Velen, your guide to traveling through the wonders of the ocean. Tonight, we're going to explore 10 little-known shark species. If these cozy moments calm you, I invite you to subscribe, share a comment, or leave a like. Now, get comfortable, take a deep breath. Don't be afraid, you're safe here. Let's dive deep into the sea and let our imagination guide us to a peaceful and deep night. There are over 1,200 species of elasmobranchs, a group that includes sharks, rays, and skates, and they are incredibly diverse, with the smallest fitting in the palm of your hand and the largest bigger than a double-decker bus. The overwhelming majority of shark species have never appeared in a documentary, have never been filmed alive, and are known to science through a single specimen accidentally caught in a trawl net. The oceans have layers. Down to 200 meters, the epipelagic zone, where 99% of the public imagines sharks live. Below that begins a system of progressively more inaccessible environments that harbors hundreds of shark species that never interact with humans, inhabiting muddy bottoms, continental slopes, underwater canyons, and abyssal plains. In this almost completely uncharted territory from a biological standpoint lives the goblin shark, Mitsucarina ostoni, the only living member of a family 125 million years old. Think of a library with over 500 books, and most of them have never been opened. That's the state of our knowledge about sharks. Science today recognizes between 400 and 500 cataloged species, members of a larger group called elasmobranchs, which totals over 1,200 distinct forms when rays and chimeras are included. Each of these species represents an independent lineage, shaped by hundreds of millions of years of selective pressure in radically different environments. Some are less than 20 centimeters long and literally fit in a closed human hand. Others exceed 12 meters and outsize any conventional road vehicle. The popular idea of a shark is built around a handful of species that inhabit shallow, warm waters: the white, the tiger, the bull. These representatives are concentrated in the epipelagic zone, the surface layer down to 200 meters. Below that boundary, sunlight gradually disappears. Between 200 and 1,000 meters, the mesopelagic zone, only diffuse twilight remains. Deeper than that, below 1,000 meters, permanent darkness sets in. Temperature drops close to 0 degrees. Hydrostatic pressure is equivalent to having hundreds of atmospheres crushing every square centimeter of structure. In this darkness of brutal pressure live dozens of shark species, completely overlooked by the public imagination. Commercial documentary cameras rarely go below 300 meters. The equipment is expensive, fragile, and logistically complicated to operate. Specialized submersibles reach greater depths, but their operating time is limited and the cost per hour is prohibitive for large-scale expeditions. The practical result is that most of the diversity of sharks remains known only through specimens accidentally caught in commercial trawl nets, dead before any behavioral observation was possible. Elasmobranchs appeared over 400 million years ago. To put that number in perspective, when the first sharks were swimming in primitive oceans, not even vascular land plants existed in their current form. Dinosaurs would still take more than 200 million years to show up. This group survived 5 mass extinctions, catastrophic events that wiped out enormous percentages of the planet's biodiversity. Every survivor carries in its biology responses to pressures that shaped the planet across entire geological eras. The bathypelagic zone, between 1,000 and 4,000 meters, harbors species with adaptations radically different from those of shallow water sharks. Flabby bodies and low musculature reflect a reduced metabolism, an essential strategy in an environment where prey is scarce and encounters are rare. Sensory organs, especially the lateral line, a system that detects pressure variations and movement in the surrounding water, become progressively more developed in deep-sea species, compensating for the absence of functional vision. Some develop hypersensitive photoreceptors capable of detecting bioluminescence from neighboring organisms. Accidental capture by commercial trawling is the main way science becomes aware of deep-sea species. Fishing vessels drag nets along bottoms between 200 and 800 meters in search of commercially valuable fish. The bycatch, unintentional capture, frequently includes sharks of unknown species. A single specimen, compressed and damaged by the rapid ascent to the surface, reaches ports and eventually laboratories. Scientists have only a few days before decomposition renders the material unusable for detailed morphological analysis. Molecular biology techniques have radically transformed the ability to recognize new species even from degraded material. DNA extracted from muscle samples allows comparison with known genomic sequences. Differences above certain percentage thresholds indicate evolutionarily distinct lineages. Species separated by enough time to accumulate characteristic mutations. This method revealed that several forms historically treated as variations of a single species actually constitute completely separate species. The total number of known species continues to grow as genetic analyses are applied to museum collections. Research aquariums rarely manage to keep deep-sea sharks in captivity for extended periods. Pressurizing tanks to the equivalent of hundreds of meters of depth requires highly sophisticated engineering. Sudden pressure drops during capture cause barotrauma, physical damage to internal organs, often fatal before any transport is attempted. The few available behavioral observations of live deep-sea species are limited to very brief encounters filmed by cameras installed in bait traps or on unmanned submersible vehicles. Biogeographic modeling applied to known distribution data suggests that the actual diversity of deep-sea sharks may be substantially greater than the number of formally described species. Entire regions of the ocean floor remain biologically unknown. Underwater canyons, structures that rival the Grand Canyon in scale, create specific habitats of high topographic complexity where endemic species can persist in complete isolation. Seamounts, with their upwelling currents that concentrate nutrients, support distinct communities that have been poorly sampled. The very concept of species becomes complex in animals whose distribution is fragmented across vast oceanic spaces. Populations separated by thousands of kilometers, with no genetic contact for enough generations, gradually diverge. The point at which that divergence justifies formal recognition as a separate species involves technical criteria debated among specialists. Goblin sharks captured in the Atlantic, Pacific, and Indian Oceans show subtle morphological differences. A 2024 genetic analysis suggested they may represent 3 distinct lineages. That single example illustrates how incomplete the map of elasmobranch diversity on this planet remains. Mitsukarina ostoni, the goblin shark, lives between 270 and 1,300 meters deep, in complete darkness. Its body is flabby, its fins are small, and its coloration is pinkish, a result of capillaries visible through its semi-transparent skin. Nothing about its anatomy suggests speed or strength, but its prey capture mechanism is one of the most extreme among vertebrates. Goblin sharks swing their lower jaw down and back to achieve a huge gape, and then rapidly project their jaws forward a considerable distance. The jaws have been recorded projecting at a top speed of 3.1 meters per second, corresponding to 8.6 to 9.4% of the shark's total body length, by far the largest and fastest jaw protrusion among sharks. The jaw launches ballistically in 322 milliseconds to snatch prey in complete darkness. In 2024, footage captured an adult swimming at 2,000 meters deep, deeper than previously believed. Imagine trying to fish in absolute darkness, with no vision, no spatial reference, in an environment where every wasted calorie could mean the difference between survival and metabolic collapse. That's the goblin shark's daily challenge. To solve that problem, the species developed one of the most extraordinary capture tools among all known vertebrates, a jaw projection mechanism that turns normally static bone structures into a variable reach instrument, like a spring-loaded mechanical arm. The basic anatomy of a shark features relatively rigid jaws positioned underneath the head. In most species, prey capture involves a direct approach followed by opening the mouth, a process that requires the predator to get within immediate range of the target before engaging it. Any prey with a fast reflex detects the approach through water displacement and escapes before contact. In open, well-lit waters, sharks compensate with swimming speed and ambush strategies. In dark depths, where a slow metabolism makes chasing costly, the solution had to be different. The goblin shark has the fastest jaws ever recorded among fish, capable of projecting out of its face like a slingshot, reaching speeds of approximately 3.1 meters per second. To visualize that speed, that's over 11 kilometers per hour concentrated in a split-second projection movement. The prey doesn't have time to process the nerve stimulus and trigger a muscular escape. The entire event happens before the victim's nervous system completes the detection, processing, and motor response cycle. The protruding jaws can extend from 8.6% to 9.4% of the animal's total body length, allowing it to catch prey that would otherwise be out of reach. In a 4-meter animal, a typical adult length, that's a projection of 35 to 40 centimeters beyond the resting position. To compare with humans, it would be like an arm that instantly extends 40 centimeters beyond normal reach, grabs the target, and retracts before any reaction is possible. The elongated, flattened snout, the species' most visually distinctive trait, is not ornamental. Its underside is covered with ampullae of Lorenzini, electroreceptory organs that detect electrical fields generated by the muscular activity of other animals. In the absolute darkness of the deep, where vision is useless and pressure disturbances can be masked by currents, electrical detection provides precise information about the location and size of nearby prey. The snout functions as a directional sensory antenna. The longer it is, the greater the detection gradient between opposite ends, allowing for more precise triangulation. The pinkish coloration that makes the goblin shark unmistakable results from the absence of dense pigmentation, combined with partial transparency of the dermis. Capillaries full of blood become visible through the superficial tissue, giving it a color that ranges from pale pink to nearly red, depending on physiological state. At depths with no light, coloration is irrelevant for camouflage or visual signaling. No predator or prey living below 1,000 meters can see wavelengths corresponding to red. The pinkish color is simply a structural byproduct with no specific adaptive function in that environment. Goblin shark teeth have an unusual morphology: thin, long, slightly backward curved, arranged in multiple rows. This configuration is optimized for catching slippery prey, fish, squid, and deep-sea crustaceans that would try to escape between less efficient structures. Once the projected jaw surrounds them, the curved teeth prevent withdrawal. The pressure during closing pushes the prey even further into the mouth. The suction generated by the rapid opening and closing of the mouth contributes additionally, creating a current of water that carries smaller particles toward the throat. A study published in 2024 in the journal PNAS revealed that, contrary to previous belief, these animals do not form a single global species, but rather 3 distinct groups, each with unique genetic characteristics. This finding has implications for the species' biogeographic understanding. Populations in the Atlantic, Pacific, and Indian Oceans may have diverged long enough ago to accumulate significant genomic differences, even while maintaining externally similar morphology. Whether these lineages constitute formally separate species remains under active investigation. Observations of live specimens are extremely rare. Most behavioral knowledge has been inferred from the anatomy of dead specimens, stomach content analysis, and an extremely limited number of recordings obtained by remotely operated vehicles. Each new piece of footage adds details previously impossible to infer from morphology alone. Swimming pattern, fin beat frequency, response to artificial light stimuli. The animal filmed at 2,000 meters in 2024 demonstrated that the known bathymetric limit needed to be revised, pushing even deeper the unexplored territory where this species lives and hunts. The reduced metabolism typical of deep-sea predators implies that the goblin shark does not need to catch prey at a high frequency. Studies of similar species suggest that meals spaced weeks apart are compatible with survival. This metabolic pattern allows for a patient waiting strategy. The animal remains nearly motionless, with minimal energy expenditure, until its electroreceptory sensors detect electrical activity from nearby prey. Only then is the jaw propulsion mechanism triggered, converting stored energy into a fraction of a second capture event. From the continental slope to the ocean floor between 390 and 1,300 meters deep lives Chlamydocoelicus anguineus, the frilled shark, an animal that the first scientists to study it, back in the 1880s, couldn't immediately identify. Its body is long and cylindrical like an eel. The mouth opens at the front end of the head, not on the underside, with a long eel-shaped body, a terminal mouth bearing 300 trident-shaped teeth, and 6 frilly gill slits that give the species its name. It is one of the most primitive sharks alive today, essentially unchanged for tens of millions of years. Its family, Chlamydocoelacidae, has a fossil record going back approximately 95 million years. All other members of that family are extinct. The gestation period is estimated at up to 42 months, the longest of any known vertebrate. The first footage of a live frilled shark dates to 2004. In 1884, Samuel Garman, a zoologist at Harvard's Museum of Comparative Zoology, received preserved specimens of an unusual fish caught off the coast of Japan. Its shape was puzzling. A cylindrical body far too long for any known shark, a mouth opening at the front of the head instead of underneath. Gill slits arranged with overlapping fleshy fringes that contrasted with the clean morphology of all familiar species. Garman recognized that he was looking at something structurally different from any elasmobranch described up to that point. He created an entirely new family, Chlamydocoelacidae, to accommodate the discovery. The name frilled describes exactly what makes this species' gills visually distinct. There are 6 pairs of gill slits on the sides of the head, each lined with expansive filaments overlapping the adjacent pairs, creating the appearance of a ruffled collar. This gill configuration is considered primitive, closer to the morphology of ancient shark ancestors than the clean, separated gills of most modern species. The scientific name itself reflects this. Chlamydocoelicus means roughly shark with a mantle, a reference to the appearance of the gill tissue. The terminal mouth, positioned at the front tip of the head rather than underneath, is another ancestral characteristic. Modern sharks developed a ventral mouth as an adaptation for benthic feeding and dive hunting. The frontal mouth suggests a lineage that predates that anatomical reorganization. There are approximately 300 trident-shaped teeth, curved inward, arranged in 25 overlapping rows, a configuration that turns the inside of the mouth into a field of inward-facing points. Any prey that enters finds no angle of escape. Every attempt to back out pushes the animal deeper into the next rows. The frilled shark's gestation period is the longest of any known vertebrate, reaching 3.5 years in estimates, based on analysis of embryos captured at different stages of development. For comparison, African elephants gestate for 22 months, already considered an extraordinarily long period among mammals. The frilled shark exceeds that by more than a year. Pups are born at 40 to 60 centimeters after developing inside the mother's uterus fed by a yolk sac, without a placenta, a reproductive strategy called ovoviviparity. This extremely long gestation period has direct implications for population viability. A female that takes 42 months to complete each reproductive cycle produces pups far less frequently than short gestation species. If adult mortality from accidental capture exceeds the replacement rate, which is already naturally low, the population declines. The depth of its habitats partially protects it from fishing, but not completely. Bottom trawling reaches the slopes where the frilled shark occurs frequently enough to accidentally capture specimens. The fossil record of the Chlamydosalactidae family reveals that the current frilled shark is the last survivor of a once diverse group. The species has a wide but extremely sporadic distribution in the Atlantic and Pacific, with most records concentrated in the western Pacific Ocean. New localities are occasionally recorded. A specimen found in southern Chile in 2025 extended the known distribution limit in the South Pacific by 850 kilometers beyond the previous record. This rarity of sightings does not necessarily indicate that the species is actually rare. It may simply reflect the difficulty of systematically sampling deep-sea habitats. The first footage of a live frilled shark dates to 2004, when cameras installed in bait traps anchored by Japanese researchers captured brief sequences of the animal in its natural environment. Previously, all behavioral knowledge was inferred. Swimming posture, prey capture method, locomotion speed were all hypotheses based on comparative anatomy. The footage confirmed some predictions and refuted others. The animal swims with body undulations of greater amplitude than predicted, resembling a snake in motion, using its entire body as a propulsive surface rather than the fins that dominate propulsion in more derived sharks. The elongated, flabby body, superficially similar to that of eels, represents functional convergence. A similar solution to a similar problem arrived at independently by completely distinct lineages. Eels are teleosts, bony fish with radically different internal anatomy. The frilled shark is a cartilaginous fish whose ancestors diverged from eels before any terrestrial vertebrate existed. The external resemblance is the product of convergent selective pressure in deep-sea environments with similar structures, not of close kinship. The frilled shark's lineage dates back to the Carboniferous period, roughly 359 million years ago, though estimates vary depending on which fossil record is used as a reference. The fact that externally very similar morphology has persisted for tens of millions of years indicates that the deep-sea environment where the species lives imposes stable selective pressures. There is no benefit in changing dramatically when the habitat remains constant. Frilled sharks found a functional configuration that solves the challenges of their niche efficiently enough to require no substantial modifications across geological eras. The depth of its distribution creates methodological challenges that make population studies extremely difficult. Estimating how many individuals exist requires density data per sampled area, information that presupposes the ability to systematically sample the habitat. For species living on slopes between 300 and 1,300 meters, that systematic sampling simply has not occurred at a sufficient scale. The frilled shark thus remains, like most deep-sea species, formally described, anatomically known, biologically inferred in many essential respects, and operationally invisible to any conservation monitoring that depends on direct observation. At 50 centimeters long and weighing 500 grams, the Icistus brasiliensis, the cookie-cutter shark, attacks humpback whales, dolphins, great white sharks, tuna, rays, and on record, the rubber sonar dome coverings of American nuclear submarines during the Cold War. The entire ventral surface, except for the dark collar, is covered by a dense network of photophores that emit a greenish glow, and that glow persists for up to 3 hours after the shark's death. The dark collar on the neck mimics the silhouette of a small fish when seen from below. A predator approaches to catch what looks like smaller prey. The thick lips and modified pharynx are used to lock onto the prey. Then the extremely sharp lower teeth bite into the skin, and rotational movements cut out a chunk of flesh. Once the plug is removed, the shark detaches, holding the fragment with its hook-shaped upper teeth and leaving behind a crater-shaped wound. Imagine a circular cutting tool barely over half a hand's length operating in the dark at hundreds of meters of depth, capable of coring out a perfect plug of flesh from a 20-ton whale, a 4-meter great white shark, or the rubber hull of a nuclear submarine. That's the daily reality of Eisischius brasiliensis, a predator that completely defies any conventional association between size and attack power. Its common name in English, cookie-cutter shark, describes the outcome of the method with surgical precision: a circular wound, clean at the edges, as if a metal instrument had removed the tissue with mechanical intent. The characteristic marks of this shark have been found on a wide variety of marine mammals and fish, as well as on the soft structures of submarines, submarine cable insulation, and other structures positioned in deep-sea regions. The list of documented victims is absurdly disproportionate to the size of the attacker. Humpback whales, common dolphins, orcas, adult great white sharks, large tuna, swordfish. None of these species is conventional prey for a 50-centimeter animal. The cookie cutter doesn't need to kill to feed. The strategy is one of active parasitism. Stealthy approach, fast strike, retreat with a tissue fragment before the larger prey even processes what happened. The shark lures prey with its glowing underside, which can deceptively resemble small fish in the deep. When the prey is close, it uses its sucker lips and upper teeth to clamp onto the larger animal, rotates its body, and cuts out a cookie-shaped piece of flesh, leaving behind the characteristic circular wound. The entire event happens in fractions of a second. The larger prey moves on. The attacker retreats with its protein portion. The cookie-cutter shark has proportionally larger teeth than any other shark, a detail that amplifies cutting efficiency. The lower teeth form an almost continuous series of triangular blades joined at the base, functioning collectively like a circular serrated edge. When the body rotates, these teeth combined with the sucker lips that maintain a seal against the prey's surface produce the precise removal of a cylinder of tissue. The resulting plug, the torn-out fragment, is swallowed whole. The wound left behind is geometric enough that early historical records mistook it for damage caused by human instruments. The incident with American nuclear submarines during the Cold War is not speculative anecdote. Even submarines have been attacked, with round chunks taken out of their sonar domes. The sonar domes, rubber covers that protect the acoustic transducers at the bow of submarines, are critical structures for navigation and detection. Damage to these structures compromises the submarine's ability to detect underwater threats, a serious problem during operations in times of intense geopolitical conflict. The U.S. Navy had to develop more resistant coatings specifically because of this half-pound predator. The cookie-cutter shark has extreme bioluminescence, with its belly covered by dense photophores that emit a strong green glow. This light emission serves a dual purpose. In the mesopelagic zones, between 200 and 1,000 meters, residual sunlight still penetrates from above at very low intensities. Organisms seen from below cast a dark silhouette against that faintly lit background. The cookie cutter's ventral photophores emit light calibrated to match the intensity of the luminous background above, eliminating the silhouette that gave away less sophisticated predators. The dark collar on the neck remains opaque, creating the illusion of a small, vulnerable fish, bait to lure the victims that become involuntary food providers. The skeleton is poorly calcified, and the disproportionately large liver functions as a reservoir of squalene, a low-density oily hydrocarbon that regulates buoyancy without the energy cost of continuous swimming. This configuration allows the animal to hover nearly motionless in the open water column, waiting for attack opportunities with minimal metabolic cost. Daily vertical migration. Descending during the day to depths exceeding 1,000 meters, ascending at night to shallower zones around 300 meters, follows the movements of potential prey that execute the same migration. Predator and prey rise and fall together, without the prey ever noticing the presence of its tiny follower. The cookie-cutter's reproduction is poorly documented because capture of pregnant females is a rare event. The species is ovoviviparous. Embryos develop inside the mother nourished by a yolk sac, without a placental connection. Litters are small. Pups are born functionally independent. Teeth are replaced periodically and in a coordinated fashion. The entire lower row replaced simultaneously, not tooth by tooth as in most sharks. This unusual pattern ensures the cutting apparatus is always in optimal condition, never compromised by gaps in the tooth series during a gradual replacement process. The species was given the scientific name Eisischius brasiliensis because it was first described from a specimen collected off the Brazilian coast in 1824 during a French scientific expedition. The geographic irony is worth noting. A shark whose attacks on American military equipment caused operational headaches was named after Brazil, the country whose coastline provided the first specimen for European science. The species' actual distribution is circumtropical, Atlantic, Pacific, and Indian Oceans in warm zones, with no special preference for the coast where it was first described. The cookie-cutter shark represents a fundamental biological principle that is often counterintuitive. Size does not determine ecological impact. Small species with specialized strategies can measurably influence populations of large predators. Characteristic Eisischius bite scars are found on significant percentages of adult great white sharks in certain populations, indicating that these attacks occur regularly enough to leave permanent records on the ocean's largest predators. A 500-gram creature leaves lasting marks on 3-ton animals, while at the same time forcing naval engineers to rethink the materials used in the most sophisticated military platforms of their era. Edmopterus perryi, the dwarf lantern shark, is the smallest known shark on the planet. It reaches a maximum known length of 20 centimeters. It is known to be present only on the upper continental slopes off Colombia and Venezuela, at depths of 283 to 439 meters. The entire animal fits in the palm of an adult hand, but it carries functional photophores on its belly and fins, capable of producing bioluminescent light. Like other members of its genus, it is capable of producing light from a characteristic set of photophores. Reproduction is aplacental viviparous, with females gestating 2 or 3 pups at a time. Curiously, some of the black spot patterns on the ventral surface are made up of light-emitting photophores, while others are made up of pigmented chromatophores. 2 different visual production systems operating side by side in the same 20-centimeter animal. Lay your hand flat on a surface and look at it. The distance from the base of your thumb to the tip of your pinky, that span, already covers the maximum length of a fully grown adult of the smallest shark on the planet. Edmopterus perryi, called the dwarf lantern shark, is the smallest known shark species, reaching at most 20 centimeters in length. It inhabits deep waters off the coasts of Colombia and Venezuela, possessing front-facing light-emitting organs likely used for hunting, reproduction, and orientation. That an animal of these dimensions belongs to the same zoological class as the whale shark, a 12-meter oceanic filter feeder, demands a revision of any mental image one has built of what a shark is. The dwarf lantern shark is known only from the upper continental slopes off Colombia and Venezuela, at depths between 283 and 439 meters. It can be identified by its small size at maturity, long flattened head, and pattern of ventral black markings and a mid-dorsal line. This extremely restricted geographic distribution makes the species vulnerable to localized habitat disturbance. A trawling fishing area that systematically covers that slope zone could capture a significant percentage of the known population in a limited number of operations. A 2021 study captured, among 153 surveyed individuals, a specimen measuring 28.9 centimeters, a value that exceeds the previously recorded maximum by nearly 50%. This discovery reopened the question of the species' true maximum size. The 20-centimeter limit may reflect insufficient sampling, not an actual biological limit. With only a few dozen documented specimens collected over decades, any descriptive statistic about the species carries substantial uncertainty. The true maximum size, life expectancy, feeding behavior, and population dynamics remain partially or entirely unknown. Dwarf lantern sharks are rare members of the genus Etmopterus, inhabit deep waters, and glow in the dark because they possess photophores generally on the belly, sides of the body, tail region, and fin edges. Photophores are highly specialized cellular structures capable of producing light through oxidation chemical reactions. In deep-sea organisms, this capability is not decorative. It serves concrete functions of communication, camouflage, and hunting in an environment where any light signal is immediately processed by eyes adapted to absolute darkness. The phenomenon that ventral photophores produce in this species, and in many other deep-sea species, is called counterillumination. The logic is the same as with the cookie-cutter shark. Any animal seen from below casts a dark silhouette against the faintly lit background created by attenuated sunlight filtering down from above. Larger predators look for that silhouette. Ventral photophores emit light calibrated to match the intensity of the luminous background, making the animal's outline virtually invisible to observers positioned below. The dwarf lantern shark optically disappears into the environment while remaining physically present, camouflaged through active light emission. Reproduction is aplacental viviparous, with females gestating 2 or 3 pups at a time. Aplacental viviparity, also called ovoviviparity, means the pups develop inside the mother's uterus nourished exclusively by each embryo's individual yolk sac, without any direct connection to the mother's circulation. This reproductive mode combines protection of embryonic development, which takes place in a sheltered environment inside the mother, with metabolic independence of each pup from the start. At birth, each offspring already has a functional version of every system it will need to survive. The 2 visual production systems that coexist on the animal's belly, light-emitting photophores and pigmented chromatophores, are structurally and functionally distinct. Photophores generate active emission. They consume chemical energy to produce photons. Chromatophores contain pigments that absorb specific wavelengths, determining passive coloration through selective reflection. Having both systems side by side on the same square centimeter of ventral surface represents a redundancy or functional complementarity whose exact purpose is still debated. The most plausible possibility: photophores regulate active camouflage against background light. Chromatophores produce recognition patterns during interspecific communication or reproduction. The dwarf lantern shark's eyes are large in proportion to its body, a classic adaptation in vertebrates that inhabit extremely low-light environments. Larger eyes capture more photons per unit of time. They function like camera lenses permanently set at maximum aperture. The retinas of deep-sea species frequently contain high proportions of rod cells, highly sensitive photoreceptors that are incapable of distinguishing colors, specialized in detecting movement and contrast in near-darkness conditions. Color vision, advantageous in well-lit environments, becomes irrelevant where all available light is monochromatic and of extremely low intensity. The discovery is so recent that the animal still has no official common name in Portuguese, an unusual situation for a formally described species, reflecting both the rarity of sightings and the scientific interest still being built around it. Most known captures occurred incidentally during research with different objectives. There is no systematic sampling protocol specifically for the dwarf lantern shark, simply because designing and executing such a protocol at 200 to 500 meters of depth off a specific South American coastline requires resources that rarely align with sufficient scientific priority to justify them. In 2010, during an expedition by the Spanish research vessel Miguel Oliver off the Pacific coast of Central America, researchers from the Smithsonian Tropical Research Institute collected 8 specimens of a completely unknown shark. A new species of bioluminescent shark was described from 8 specimens collected off the Pacific coast of Central America, at depths between 836 and 1,443 meters. Researcher Vicky Vasquez asked her 8-year-old cousins and a group of high school students to suggest a common name for the species. The suggested name, Ninja lantern shark, refers to the uniform black coloration and the reduced set of photophores used for camouflage in this species, somewhat reminiscent of the typical outfit and stealthy behavior of a Japanese ninja. The scientific name, Itmopterus benchlei, honors Peter Benchley, author of Jaws and marine conservationist. 8 specimens, that is the total volume of biological material on which the entire scientific description of a brand new species was built. The ninja lantern shark inhabits the eastern Pacific Ocean, from Nicaragua to Panama and Costa Rica, at depths between 836 and 1,443 meters along the continental slope. It is the only species of the genus Itmopterus known off the Pacific coast of Central America. That an entire species occupies a restricted geographic region and remained completely unknown until 2010, decades after scientific submersibles began operating systematically, illustrates just how superficially the deep ocean has been explored to this day. The species was described from 8 specimens collected during an expedition by the Spanish research vessel Miguel Oliver, led by researcher D. Ross Robertson of the Smithsonian Tropical Research Institute. The vessel was conducting biological surveys of species of commercial and scientific interest. The ninja lantern shark was an incidental catch, not an intentional target of the expedition. This pattern of discovery by accident is the norm in deep-sea taxonomy. New species are rarely found by expeditions specifically designed to find them. They turn up as surprises in nets whose primary objective was something else entirely. The privilege of suggesting a common name for the species was granted to 4 young shark enthusiasts between 8 and 14 years old, relatives of the study's lead author. The decision was not a whim. It reflected an explicit philosophy of connecting science with the community, specifically in honor of Peter Benchley's legacy, whose career was marked precisely by turning popular interest into conservation engagement. Involving children in naming a new species made that connection between young audiences and still uncatalogued ocean biodiversity tangible and real. Researcher Vicky Vasquez explained to the children that the shark uses photophores in its skin to produce a faint glow in the dark ocean. The combination of that stealthiness with a completely black appearance led the children to suggest the name Super Ninja Shark. Vasquez felt the longer version would hinder adoption by the scientific community. Common names for species tend to converge on concise, descriptive forms. The shortened version, Ninja lantern shark, preserved the central element of the children's proposal while following a nomenclatural convention more compatible with scientific literature. Ninja lantern sharks have uniformly black skin, in contrast to the gray and brown tones of other known lantern sharks. Another distinguishing feature is the number and arrangement of photophores across the body, concentrated mainly on the head and fewer in number compared to other members of the genus. This unusual photophore configuration is key to understanding the function of bioluminescence in the species. While most lantern sharks cover the entire belly with photophores for counterillumination, the ninja appears to use its light in a more selective and localized way. Hence the hypothesis of partial camouflage, rather than complete silhouette elimination. Researcher Vicky Vasquez explained that the fluorescence ability may function as a form of camouflage. The principle is counterillumination. The shark glows just enough to eliminate its shadow when seen from below, without becoming conspicuous through excess light emission. This fine-tuned regulation of light intensity, not dark enough to cast a silhouette, not bright enough to be detected by light-sensitive predators, requires precise control of photophore output. How that control is physiologically exercised is still under investigation in more thoroughly studied species of the genus. The scientific name Benchley honors Peter Benchley, author of Jaws and later an active champion of ocean conservation. His legacy includes the Benchley Awards, which recognize outstanding achievements in marine conservation. Benchley's biographical irony is well documented. The same writer whose 1974 novel fueled decades of irrational fear of sharks became one of the most active public advocates for the conservation of these species. Naming a new species after him connects the beginning, the novel that distorted public perception, with the end, the conservation legacy that tried to undo part of that damage. Very little is known about the ninja lantern shark. Researchers still don't know what it eats, what threats it faces, or how widely distributed it is. With 8 specimens as the entire knowledge base, any behavioral inference is highly speculative. Analogy with closely related species of the same genus provides working hypotheses. Feeding on invertebrates and small fish, daily vertical migration, bioluminescent communication during reproduction. But no direct observation confirms any of them for this specific species. The process by which a new species goes from 8 frozen specimens to a formally described and named taxon involves comparative morphology, analysis of meristic characters, counts of vertebrae, spines, fin rays, and increasingly genetic sequencing for confirmation. About 20% of known shark species have been discovered in the last 10 years, according to one of the study's researchers. Our job is to look for these lost sharks. That rate of discovery, in a group of relatively large vertebrates that have been studied for centuries, is direct evidence of just how incomplete the inventory of deep-sea ocean life remains, and of how many species are still out there, in the dark, waiting to be found. In November 1976, a naval anchor dropped off the coast of Hawaii at 165 meters deep snagged an animal that the researchers on board had never seen before. A 4.5-meter shark with a massive, wide, rounded mouth positioned at the front of the head instead of on the underside. It was Megachasma pelagios, the megamouth shark. Fewer than 100 confirmed specimens have ever been seen or captured worldwide, making it one of the rarest known sharks. In 2024, tagging studies in the Northwest Pacific confirmed that megamouth sharks migrate vertically, staying in the depths during the day and rising at night to feed on plankton. Typically found between 500 and 1,000 meters during the day, each sighting is treated as a scientific event. Its reproductive biology, social behavior, and global distribution remain practically unknown. The sequence of events was so unexpected that the first specimen took 7 years to be formally described. On November 15, 1976, a team of scientists from the University of Texas was testing a sonar system aboard the vessel AFB-14 from the Naval Ocean Systems Center about 42 kilometers off the coast of Oahu, Hawaii. While hauling in the equipment after nighttime tests, they encountered unexpected resistance. And emerging from the depths tangled in one of the sea anchors, a large shark over 4 meters long. None of the scientists on board could identify the species. The morphology simply didn't match anything in existing records. When examined by specialists, the specimen turned out to be not just a new species, but an entirely new genus and family, later named Megachasmidae. The creation of a new family is an extraordinarily rare event in vertebrate taxonomy in the 20th century. Most discoveries fill gaps within already established families. The fact that a 4.5-meter shark, living in oceans traveled by vessels for centuries, had completely escaped scientific knowledge until 1976 reshaped expectations about what might still be hidden in the deep. The name Megachasma means enormous open mouth, and pelagios refers to living in open water, away from the ocean floor. That etymology captures the two most relevant characteristics of the species: the disproportionately large mouth relative to the body, an adaptation for filter feeding on plankton, and the habit of inhabiting the open water column rather than the ocean floor. Other giant filter feeders, such as the whale shark and the basking shark, also have wide terminal mouths. The megamouth independently converged on a similar strategy from an evolutionarily distinct lineage. The megamouth shark has photophores around its mouth, light-emitting structures that likely attract zooplankton and small crustaceans into the oral opening as the animal swims with its mouth open. Bioluminescence in that anatomical position functions as a light trap. Organisms that orient toward light in the dark depths are funneled directly into the filtration apparatus. This mechanism has not been directly observed in the field, but is supported by the position of the photophores and the documented nighttime vertical ascent behavior. Since its discovery in 1976, fewer than 280 animals of this species have been seen worldwide. That number represents all specimens captured, found dead on beaches, and observed alive across nearly 50 years of global records. For comparison, a single school of hammerhead sharks or a whale shark aggregation can involve hundreds of individuals in a single location on a single day. The rarity of megamouth sightings is not a product of behavioral shyness. It reflects dispersed distribution across inaccessible deep-sea habitats. A specimen measuring 5.3 meters was found stranded on Praia Grande Beach, in Arraial do Cabo, Rio de Janeiro, representing the 43rd known individual in the world and only the third recorded in the Atlantic. The necropsy indicated that the animal appeared to have died from natural causes, with a completely empty stomach. The fact that the third Atlantic record of the species was found in Brazil demonstrates that the megamouth's distribution includes the South Atlantic, expanding the geographic area where this shark can be expected beyond the concentrations in the Western Pacific. The documented daily vertical migration, descending to depths between 500 and 1,000 meters during the day, rising at night to shallower layers, is a strategy shared by many deep-sea species and responds to the movements of the prey themselves. Zooplankton and small mesopelagic crustaceans execute the same migration. They descend to escape visual predators during the lit period and rise at night when darkness offers relative protection. Predators that follow this cycle ensure constant access to prey without spending energy on active searches across a vast three-dimensional environment. The absence of information about megamouth reproduction is almost total. No pregnant female has been examined with identifiable stage pups. The reproductive mode, oviparous, ovoviviparous, or viviparous, is inferred by analogy with close relatives, not by direct observation. Litter size, gestation period, sexual maturity, and longevity are completely unknown. This gap is relevant for conservation. Without understanding the reproductive rate, It is impossible to assess how many individuals die from accidental capture before reproducing, and whether that mortality exceeds the population's replacement capacity. Every new specimen captured or cited generates a scientific publication. This protocol of systematic documentation, unusual for abundant species where individual records rarely justify publication, reflects the status of extreme rarity. Every data point is valuable because the total available dataset is small. When a megamouth shark is found alive, researchers mobilize resources to sample tissue, take precise measurements, photograph anatomical details, and when possible, implant tracking tags. The animal is then released, carrying instruments that transmit location and depth for weeks before the batteries run out. Echinorinus coucae, the prickly shark, is covered in spine-shaped dermal denticles that dot its entire body like armor made of nails. It can reach 4.5 meters in length. It is a rare deep-water species found from 11 to 1,100 meters deep in the western Pacific Ocean, off Japan, Taiwan, Palau, Australia, and New Zealand, and in the eastern Pacific off Hawaii, the Gulf of California, and from Costa Rica to Peru and Chile. Despite its size and wide distribution on paper, sightings are so scarce that the IUCN classifies the species as data deficient, without adequate information to assess extinction risk. In May 2022, 12 prickly sharks were spotted at 151 to 350 meters deep on the seamounts of the Coiba Ridge in Panama, the first documented evidence of live specimens of the species in that region. 12 individuals together, the largest group ever recorded in the history of this species. Imagine covering a 4-meter shark with hundreds of metal nails distributed uniformly across the entire body surface. Each nail curved sharp, robust enough to deter any predator from biting into its flank. That is the functional description of the dermal denticles of Echinorinus coucae. Not the smooth, aerodynamic denticles that cover fast sharks like the mako, but calcified spine-shaped structures projecting visibly above the skin surface, turning the animal into a surface no predator with conventional teeth would easily bite into. The genus Echinorinus includes only 2 living species of unusual and poorly known sharks. Both are relatively large, between 3 and 4 meters in length. They are characterized by a short snout and spine-shaped dermal denticles, some of which may be fused. The phylogenetic classification of the family is debated. Morphological and molecular analyses place the group sometimes within squaliforms, sometimes as a sister group to angel sharks or saw sharks, an unstable systematic position that reflects transitional morphology between groups that diverged long ago. In May 2022, 12 prickly sharks were spotted at depths between 151 and 350 meters on the seamounts of the Coiba Ridge in Panama. The discovery expands knowledge about the distribution and habitat use of this rare species and highlights the potential importance of the Coiba Ridge seamounts as critical habitat for it. Seamounts, elevations of the ocean floor that don't reach the surface, are structures that deflect upwelling currents, concentrating nutrients and creating conditions that support denser biological communities than the flat surrounding floor. They serve as oases across stretches of relatively barren seafloor. The publication represents the first documented record of live Echinorinus coucae specimens in Panama, although unverified reports previously existed about its presence in the tropical eastern Pacific. The distinction between an unverified report and a documented record is fundamental in taxonomy and biogeography. Science operates on confirmable observations, not on fishermen's rumors or anecdotal accounts without associated physical material or footage. That a 4-meter species inhabited Panamanian waters without formal confirmation for decades of underwater scientific exploration of the region demonstrates just how fragmented scientific coverage of deep-sea habitats remains. The figure of 114 pups per litter attributed to the species in early records requires context. Species of the genus Echinorinus are ovoviviparous, with litters of up to 24 pups documented. The figure of 114 circulates in some popular sources, but derives from a single specimen analyzed with an exceptional number of embryos, possibly a counting error, an individual anomaly, or a fusion of counts of ovarian structures at different stages. Litters documented in other records are substantially smaller. This is precisely the type of uncertainty that persists when a species is known from so few specimens. A single atypical individual can skew all available statistics. The IUCN's data-deficient classification is technically distinct from threat classifications like vulnerable or endangered, but it does not mean the species is necessarily safe. It means the available data are insufficient to determine the risk, which may be high or low without any way to tell. For deep-sea species caught as bycatch in trawl and longline fisheries, the absence of monitoring data does not imply the absence of impact. It implies the absence of monitoring. That distinction is relevant for conservation decisions that depend on evidence to justify fishing restrictions. The aggregative behavior suggested by the sighting of 12 individuals together in 2022 is biologically significant information. Solitary sharks and social sharks require completely different management strategies. If the species aggregates at specific locations, such as seamounts, during particular periods, those aggregations may coincide with reproductive activity, the phase of greatest vulnerability to capture. Protecting aggregation sites would be more efficient than attempting to regulate fishing across the species' entire geographic range, which stretches for thousands of kilometers of Pacific coastline. The spiny dentical skin, beyond its defensive function, may also play a role in hydrodynamics. Rough surfaces interact with water flow in ways that smooth surfaces do not reproduce, creating microturbulences that under certain conditions reduce drag. Slow-moving deep-sea sharks, less concerned with top speed than with long-term energy efficiency, may benefit from surface characteristics different from those that optimize fast swimming. This hypothesis has not been directly tested on the prickly shark, but is compatible with patterns observed in other slow-moving species with irregular skin textures. No specimen of Echinorinus cucae has survived in captivity long enough to allow systematic behavioral observation. Attempts to keep captured individuals in large aquarium tanks resulted in relatively rapid death, a common pattern in deep-sea sharks that cannot tolerate the pressure, temperature, and water chemistry conditions maintained at surface facilities. All available behavioral knowledge comes from short-duration footage obtained by remotely operated vehicles during accidental encounters, and from the 2022 sighting at the Kuiper Ridge, which represented, in a single event, more observation of aggregative behavior than all prior literature combined. Oxynotus centrina, the angular rough shark, has a body that looks like no other shark. Its body has a distinctively curved, almost pyramidal appearance, due to its tall triangular dorsal fins and robust, compressed torso. The skin is covered in denticles so prominent it feels like coarse sandpaper. It lives between 60 and 1,300 meters deep in the eastern Atlantic and Mediterranean. One of the most fascinating characteristics of the angular rough shark is the sound it produces when brought to the surface by fishermen, a low, grunting noise, possibly due to the release of air or water from the body. Angular rough sharks are not commercially fished. It is believed they bring bad luck to the fishermen if caught and kept. Unfortunately, when released, they have only a small chance of survival. In 2024, the first documented case of leucism in the species was recorded off the coast of Albania, an individual with abnormal coloration, partially depigmented. Sharks don't grunt. That statement holds true for virtually all of the nearly 500 species in the group. cartilaginous fish with no swim bladder, no specialized sound production mechanisms, no internal air chambers that would allow acoustic emission. And then there is Oxynotus centrina, which breaks that rule too. The sound it emits when pulled from the water by fishermen has been described as a low grunt, an unlikely vocalization in a group where silence is the norm. The exact mechanism has not been fully explained, but hypotheses include muscular contractions forcing water through narrow gill passages, or compression of bodily fluids during handling that produces an audible sound as they escape through specific openings. The angular rough shark inhabits marine waters in the eastern Atlantic from Norway to South Africa, at depths of 40 to 780 meters. This extensive latitudinal distribution, covering temperate and subtropical zones of both hemispheres along the eastern Atlantic, coexists with a rarity of sightings that indicates low population density or behavior that systematically avoids the environments where scientific sampling occurs most frequently. Species of low density and wide distribution present specific monitoring challenges. No single area concentrates enough of the population for efficient sampling. The angular rough shark's silhouette is immediately recognizable to anyone who sees it once. With a small, robust body, wide-set eyes, a pinkish snout, large nostrils, and distinctive dermal scales, the animal resembles a cross between a shark and a caricatured pig, hence the informal nickname pig shark in some languages. The triangular dorsal fins, disproportionately tall relative to the body, give it a pyramidal profile when seen from the side. This laterally compressed body morphology, as if the animal had been squeezed from both sides, is an adaptation to a demersal lifestyle, gliding over irregular ocean floor substrates without any need for speed. The angular rough shark is generally dark grayish-brown or even black in color, with dark spots on the head and sides. This dark coloration is important in helping them blend into their environment as an example of mimicry. On muddy or rocky bottoms at moderate depths, where the species hunts polychaetes, crustaceans, and bottom-dwelling fish, dark coloration and rough skin texture produce an efficient combination of passive camouflage. The motionless animal on dark substrate becomes difficult to detect both for prey that might escape and for predators that might threaten it. The belief that catching this shark brings bad luck to the fishermen is not simply superstition without a practical basis. There is underlying pragmatic logic. The animal has no commercial value, is difficult to handle due to its abrasive skin, and produces a disturbing sound that contributes to discomfort during handling. Developing a reputation for bringing misfortune functions as a cultural mechanism that discourages fishermen from keeping specimens, and that, inadvertently, protects the species from additional pressure on an already scarce population. The bad luck is informal conservation encoded in fishing tradition. In 2024, the capture of a rare, critically endangered adult with abnormal coloration was reported near Sazan Island, Albania. The shark displayed partial reduction in pigmentation, resulting in a pale appearance with whitish-gray patches. Since retinal pigmentation appeared to be normal, the specimen was classified as leucistic. Leucism differs from albinism. Albinos lose melanin completely, including in the eyes, resulting in impaired vision due to the absence of retinal pigment essential for light absorption. Leucistic individuals lose pigmentation only in the skin and external structures. The eyes retain functional pigmentation and normal vision. The study published in October 2024 in the Journal of Fish Biology represents the first description of leucism in angular rough sharks and the first record of a pigmentation disorder across the entire family Oxynodidae. This double first time in the species, first time in the family, reflects both the rarity of the phenomenon and the rarity of sightings of the species itself. For a pigmentation disorder to be formally documented, one must first capture a specimen, correctly identify it, and have researchers present or accessible to examine and publish the finding, an unlikely sequence in a species that is already rarely observed. The lack of pigmentation may make individuals more visible to both predators and prey, potentially reducing the leucistic animal's chances of survival. For a species that depends on passive camouflage as its primary hunting and evasion strategy, losing cryptic coloration is a concrete functional disadvantage. Prey spot the predator before being caught. Larger predators detect the leucistic individual before escape strategies can be executed. In a species with already reduced abundance, each individual represents a significant fraction of the local population. The loss of any adult carries proportionally greater demographic weight than in numerically abundant species. Research indicates that leucism may have varied causes, including genetic mutations, exposure to pollutants, elevated temperatures, and hormonal imbalances during embryonic development. The diversity of potential causes makes it difficult to attribute specific cases to a single mechanism without detailed genomic analysis. For deep-sea species inhabiting environments far removed from direct sources of industrial pollution, spontaneous genetic mutation or embryonic hormonal disruption are more plausible hypotheses than direct environmental contamination. The record of this particular individual, often Albanian coast, that is emerging as a location of interest for endangered Mediterranean species, adds yet another layer of conservation relevance to the sighting. From the goblin shark that fires its jaw at 3.1 meters per second in the dark to the angular rough shark that grunts when caught, from the smallest known species, the 20-centimeter dwarf lantern shark, to the megamouth shark accidentally discovered on a naval anchor in 1976, the unknown sharks of the oceans share one characteristic. Science knows them poorly, superficially, or barely at all. The ninja lantern shark was formally described in 2015 from 8 specimens. The prickly shark had the largest group ever recorded documented in 2022, 12 individuals. The cookie-cutter shark attacked submarines for decades before being identified as the culprit. The frilled shark was filmed alive for the first time in 2004, more than a century after its scientific description. Sharks have existed for 450 million years, surviving 5 mass extinctions, older than dinosaurs and even older than trees. And yet, despite all that, most of their species are still more unknown than known. 450 million years. To put that number in perspective, it helps to build a mental scale. If the entire history of sharks were compressed into a single calendar year, the first dinosaurs would only show up in September. The first trees would appear in July. The first anatomically modern humans would arrive in the final hours of December 31st. All of the scientific knowledge accumulated about sharks, including the discovery of the megamouth, the filming of the frilled shark, the description of the ninja, would happen in the last seconds of that imaginary year. A group that has crossed 500 million years of planetary history has been systematically studied for only the blink of an eye on a geological scale. Each of the species covered in this narrative represents a distinct category of scientific ignorance. The goblin shark is known anatomically but behaviorally inferred. Its prey capture was filmed by accident, not by experimental design. The frilled shark spent over a century as a preserved corpse before being observed swimming. The megamouth was discovered only because it got tangled in the anchor of a military vessel. Without that specific accident, the species could have remained unknown for decades more. The ninja lantern shark exists as a scientific concept based on 8 frozen specimens. The prickly shark had its largest ever documented group recorded at 12 individuals. The pattern that emerges from these individual stories is systematic, not accidental. The ocean depths are inaccessible not for lack of scientific interest, but for concrete physical and economic reasons. Water pressure increases at a rate of approximately 1 atmosphere every 10 meters of depth. At 1,000 meters, a depth where several of these species routinely live, the pressure is 100 times greater than at the surface. Building and operating equipment capable of functioning under those conditions costs as much as space exploration. 1 hour of scientific submersible time costs more than most annual budgets of marine biology labs. Genetic tracking technology has radically transformed the ability to recognize new species without capturing them alive. Environmental DNA, genetic fragments left by organisms in the surrounding water through skin cells, mucus, and excretions, can be extracted from water samples collected by simple diving equipment. Comparison with growing genomic databases allows the presence of species to be detected without any individual being seen or captured. Early applications of this technique in deep-sea habitats have already revealed genetic signals of formerly undescribed species. Organisms present in sampled ecosystems whose existence is only now beginning to be inferred. The last mass extinction that sharks survived, the Cretaceous-Paleogene event 66 million years ago, which wiped out the non-avian dinosaurs and approximately 75% of all animal species then in existence, did not eradicate the group. Survivors repopulate progressively impoverished oceans, diversifying again over millions of years. This historical resilience is frequently invoked as an argument that sharks are untouchable, that nothing can truly threaten them. That argument confuses long-term evolutionary durability with invulnerability to current short-term pressures. Estimates of annual shark deaths from human activity range between 70 and 100 million individuals. Intentional capture for fins, bycatch in nets aimed at other fish, recreational fishing, and vessel strikes. For species with slow reproduction, late maturity, and small litters, like the frilled shark with its 42-month gestation or the angular rough shark with 7 to 12 pups, that mortality can far exceed the natural replacement rate. The difference between a group that survived 5 mass extinctions and a group that collapses under human pressure is speed. Geological extinctions occurred on timescales that allowed some adaptation. Current pressures operate in decades. The incomplete inventory has a direct practical consequence. It is impossible to protect what you don't know exists. Marine conservation policies depend on species lists and status assessments to set priorities, create protected areas, and regulate fishing. When species like the prickly shark are classified as data deficient, they don't enter active protection frameworks. Not because they are safe, but simply because there isn't enough data to justify any risk classification. That epistemic gap is itself a threat, just as real as trawl nets or fin fishing. The discovery of the megamouth shark in 1976 was 50 years ago. The first footage of a live frilled shark was just over 20 years ago. The ninja was described only a decade ago. Each of these timestamps suggests that the calendar of discoveries is not close to closing. It is accelerating as more accessible technologies and more sensitive genetic methods reduce the cost of exploration. Moderately priced expeditions equipped with low-cost deep-sea cameras, remotely operated vehicles accessible to smaller institutions, and environmental DNA techniques are democratizing research in habitats that once required the resources of only the world's largest institutions. The goblin shark that opens this narrative, firing its jaws in the dark at 3.1 meters per second, inhabiting depths where sunlight has never reached, carries within its existence the synthesis of everything this collection of species represents. A planet still partly unknown, inhabited by organisms with biological solutions that no human mind could have anticipated without encountering them directly. Every undocumented species is both a gap in knowledge and a promise of discovery. The darkness below 200 meters is not empty. It is incognita in the original sense of the word. territory that exists, but that still waits to be known by those with instruments capable of reaching it.